Today we are going to take a seminar on the topic RNA polymerase of prokaryotes. In molecular biology, RNA polymerase, or more specifically DNA-directed or dependent RNA polymerase, is an enzyme that synthesizes RNA from a DNA template. Most prokaryotes, single RNA polymerase transcribes all types of RNA. Transcription process is catalyzed by enzyme DNA-dependent RNA polymerase and was discovered by Samuel B. Weiss and Gerard in 1916. In prokaryotes, only one type of RNA polymerase is responsible for the synthesis of different types of RNA, such as tRNA, mRNA, and rRNA. Now, to the structure of the RNA polymerase. In E. coli, the polymerase is composed of 5 polypeptide subunits, 2 of which are identical, and they are 2 alpha, beta, beta dash, and omega. These polymerase composed of 5 subunits is together called a core enzyme, and each of the subunit has a unique role. The alpha subunit is necessary to assemble the polymerase on the DNA, and the beta subunit binds to the ribonucleoside triphosphate that will become a part of the recently bound mRNA molecule, and the beta dash bind the DNA template strand, and the sigma factor is only involved in the transcription initiation. Core enzyme plus sigma factor is the hollow enzyme. Without a sigma factor, the core enzyme transcribed from random sites and could produce mRNA. Now to the transcription in prokaryotes. Transcription is the process of RNA synthesis from DNA. It involves mainly 3 steps. They are initiation, elongation, and termination. To the first step, initiation. The binding of the RNA polymerase to the DNA is prerequisite for the transcription to start. The specific region on the DNA where the enzyme bind is known as a promoter region. The structure and function of the prokaryotic promoter is relatively simple. One important sequence in prokaryotic promoter is located in 10 bases before the transcription start site, and it is commonly called data box. -10 and -35 region upstream the initiation site. Now to the second step, elongation. In elongation, the RNA polymerase recognizes the promoter region, and sigma factor is released and transcription proceeds, and the RNA was synthesized from the 5' end to the 3' end. DNA is continuously unwound ahead of the core enzyme and rewound behind it. RNA polymerase acts as a stable linker between the DNA template and RNA strand to ensure that elongation is not interrupted. And now to the last step, it is termination. The process of transcription stops by termination signals. They are of 2 types. They are row dependent termination and row independent termination. In row dependent termination, specific protein named row factor bind to the growing RNA, and in bound state it acts as ATPase and terminates the transcription and release RNA. It was also responsible for the dissociation of RNA polymerase from DNA. Then to the row independent termination. Termination in the case is brought about the formation of hairpins of newly synthesized RNA. This cause is due to the presence of palindromes. Thank you.